Good evening to everyone present. I, Shresh Singh, am present here to today welcome you all to day 3 of Cognizance, an interdisciplinary event organized by the Research Society MIT. Cognizance is an initiative which hopes to bring together all people interested in research in various fields. Our organization is confident that this talk will provide value to everyone who is attending. Today we have with us Dr. Subrahmanyam Vangala to enlighten us with his knowledge, and we have Manthan Joshi, Biotechnology Expertise Head at RSM, who is going to introduce the speaker and moderate the session. Before the presentation begins, I would like to define the structure of the talk today. Our speaker will be giving a presentation on the topic, followed by a question and answer session. Throughout the talk, the chats will be open to discussion for all the attendees. You can put up your questions there, and we will be asking them to sir at the end of the presentation. Our team will be moderating the chats. We would request everyone to keep the contents of the chat related to the topic. Having said that, I would like our moderator to introduce the speaker for the day. Over to you, Manthan.
Good evening and welcome to Cognizance, presented to you by the Research Society MIT Manipal. Today's speaker, Dr. Subrahmanyam Vangala, known as Mani, is currently the CEO and co-founder of Reagene Biosciences based in Bangalore, India. He received his PhD in biochemistry at Memorial University, Canada, followed by a postdoctoral training at University of Toronto, University of Colorado, and University of California at Berkeley. He has over 30 years of pharmaceutical industry experience in USA and India at many companies including Viat, J&J, and Purdue Pharma. His research is focused on discovering and developing novel therapeutic agents for many diseases, including cancer, diabetes, insomnia, and anti-infectives and pediatric genetic abnormalities. He and his colleagues together conducted extensive research and helped market at least seven novel drugs. His talk today will focus on a bird's eye view evolving around the paradigms of the on overall drug discovery and regulatory development process, paving the way to personalized and precision medicine for each and every patient. Now, I shall hand over the stage to Dr. Subramaniam Vangala. Thank you.
Yeah, can you guys hear me? Hello?
Yes, sir. Yes, sir.
Yes.
Okay, great. Thank you, Manthan, and I'm really honored to be here today to give some. I think most of the people here are students and trying to be future scientists of India, and I think I'll try to keep it as non-technical as possible for people to see what exactly is happening in drug discovery over the years around the world, especially in the US, where I worked for nearly 3 decades. But the thing is that drug discovery is a very complex process, and it's not easy to discover drugs. And I'm going to giving you some bird's eye view of the challenges that we face. And in fact, the productivity is always low because of the complexities of the biology that we need to unravel. So I'm putting up my slides here right now, and can you guys see the slides?
Yes,
Hello?
sir, they are visible. Yes, sir, they are visible. You can continue.
I can't see them right now. Okay, got it. Okay, so the drug discovery I've been involved since 1992, early 1990s. It's been a challenging experience for the last 30 years. And the interesting thing is that every year you find new ways of doing things. It's not just a model that you use for all drugs or anything. It's not something that you can actually discover a drug overnight. It takes about uh, maybe 10 to 12 years or more in some cases. For example, for Alzheimer's disease, so far we don't have any drugs really that can treat. Although recently one drug from Biogen has been approved by FDA, but we still don't know whether it really works in the patients or not. So the drug discovery, especially if you go to cancer and neurodegenerative diseases, there are not that many drugs out there. It's very challenging to discover drugs in those areas. Where we have been successful is antibacterial drugs and other small diseases that maybe gastric acidity, headaches. These are the things what we have. The majority of the market is filled. Diabetes, of course, has got lots of drugs and some. Yeah, lots of drugs are there for diabetes as well. And so what we need to do is the journey of the drug discovery, where it starts and where it ends. Uh, Next slide. Okay. So what is the criteria, two major important criteria for approval of a drug by regulatory agencies? I mean, what regulators are afraid if the drugs can kill patients and which they don't want to. So they pay more attention to the safety than whether the drug works. It's a reality. I mean, FDA also, yeah, if there's some marginal efficacy, they'll approve it, but as long as it's very safe. And if you look at it, you can think of it as a traffic light approach. Uh, if the drug is very safe and it actually works very well, like maybe 80% of the people population, then it's no brainer. It's a green, like a traffic light, you just can pass through it. But if the drug has good efficacy but very narrow safety margin, then you got to look at it like a proceed with a caution, yellow light. And if the drug has more risks, adverse events, especially fatal drug-drug interactions and other parameters, you got to stop right there, red light. And this is how we represent in the next slide I'll show you, is that if I have a 12 drugs with that lead candidate, right? I have various models that I put into it, and here the different experiments we do, I put them in a table. What all I do is looking at the liabilities, I put the colors on the for these 12 drugs and say which drug is the best. You see the candidate number one, all greens. Okay, go take this one into the clinical trials. But they're all red drugs. We don't take them into the clinical trials because they're too much of a liability to go into the clinical trial. And if there are greens split with some yellows, yeah, it's okay. I mean, you can still go with them, but more greens are there. So yellows, if you're manageable, then you can go ahead with it. So this is how we actually look at a green, red, and yellow strategy for drugs go no go decisions. We have plenty of diseases in the world. I mean, you know what? This is the world that right now highly polluted, a lot of infectious diseases, and today we eat only McDonald's and Kentucky Fried Chicken, the pizzas and stuff. And so, with the modern world, the diseases are a different kind compared to maybe 100 years ago, 200 years ago. Today, the top 10 diseases that you can see is that this is from 2015. Uh, Top 10 diseases, ischemic heart disease and coronary heart disease tops the list, with 8.8 million people died with ischemic heart disease. Then followed with stroke, lower respiratory tract infections, chronic obstructive pulmonary disease, trachea, bronchus, and lung cancers, diabetes, Alzheimer's disease, dehydration. This is very interesting. Dehydration due to diarrheal disease. Okay. I never thought this would be in the top 10, but it is. And tuberculosis. Tuberculosis is still prevalent in the third world countries. In the US also there is some emergence of tuberculosis, but we have all these developing world has has more tuberculosis. And cirrhosis. Cirrhosis is like uh, it typically represents the liver disease, and it's also due to the alcoholic cirrhosis, etc. A lot of people drink now. Too much drinking can cause cirrhosis. So that's also in the top 10 diseases. So about 30 million people died with all these top 10 diseases. Apart from this, the world is filled with orphan diseases. There are 7,000 orphan diseases. And each disease, you classify orphan disease as less than 200,000 people are affected in the world. So the population is too small for the orphan disease. If I want to make a money making a drug out of this orphan disease, it's too tough to make money out of it. Although, I mean, if you're philanthropic and if you have a lot of money, you want to spend it, and you can work on these orphan diseases. But in fact, FDA has given some criteria. Hey, this is orphan disease. We have some special approval process for you if you come with, because these are unmet medical needs. There's no drug out there. There's really important that a drug should be there for these people. In those cases, FDA says that okay, instead of spending 10 to 12 years, will approve a drug with 4 to 5 years with the data you have and the clinical data. So there are some things you can negotiate with the FDA for orphan diseases. But all these orphan diseases are actually, most of them are genetic defects, born genetic. So how does the drug discovery process grow? If you look at in big pharma companies, uh, they screen millions of compounds on the computational modeling, and then narrow it down to 10,000 to 20,000. Some chemists may not synthesize all these compounds, but they further narrow it down with specific properties to 400 to 500 manageable candidate drugs. So they're not drugs yet. You can't call it drug unless it has phase 2 proof of concept in the clinic. So these are candidate drugs that have drug-like properties. So you screen them all through like a funnel. You filter them out at each step, and you have lead optimization stage that you can screen out a lot of compounds, 90% of these compounds, and then you have ADME and toxicology is another step where we actually do more screening process. Then you go into the first in human application, which is called IND, investigational new drug. And that allows us to go into the clinical trials. There's a lot to be done in toxicology. They're all not just doing toxicology in animals. There's a lot of GLP, GCP, compliance activities, regulatory activities that we need to be aware of how we do it, because the documentation is so important to go into enabling studies. Once market, it may cost about $2.5 billion, and you have 10,000 to 20,000 candidate drugs. You're coming down to one drug to market. So it's a very risky business. I mean, I'll never know. I mean, spending 15 years, I'll get a drug or not at the end of the day. So you're spending $2.5 billion. It's a big liability, financial liability. So if you look at the historical USA drug approvals, it has been always between 40 and maybe 2015, etc. And if you look at that, big pharma companies, the blue line, you can see that from 1996 to 2004, those are the big pharma approvals. But if you look at small biotechs, which are actually the yellow part, are also getting more approvals than the big pharma. So more science is conducted in small biotech companies than in big companies, big pharma companies. And if you look at the chart, the poor productivity, 19, you can say 2000 to maybe 2015, etc., the drug approvals become very, very tough to get. I mean, you can see maximum 25 drugs got approved. Although in 1996, you can see 53 drugs approved. That was exceptional. And not until 2018, we got the chart back up to 59. Then you have again 2019 down to 48, 2020, 46. 2021 till June, we have only 20 drugs approved. I'm not sure we'll reach the 59 or 53 mark in 2021 either. So yeah, it's always flat. If you look at it, the majority of the drugs, maybe around 30 to 40 drugs are approved per annum. Maybe some of them are new chemical entities. Some of them are actually biological, so protein molecules, antibodies, etc., etc. And also some orphan drugs too. In the recent years, orphan drugs have been approved quite a lot. Here it is biological sources, new molecular entities, and you can see in 2018, I think, you have increased approvals of the biologicals. So 17, so there's a lot more and more biologicals than just small molecules are being approved by the FDA also. And look at the orphan drugs. In fact, I don't have the exact number, how many orphan drugs were approved per year, but if you look at the sales wise, orphan drugs account only 10% or 9.6% sales compared to the entire drugs that are approved in the market. About $451 billion sales for the drug market in 2017, out of which orphan drugs only accounted for 10% of the sales. But interestingly, that orphan drugs are also used for non-orphan indications in some cases, which is actually more higher market. You see $69 billion was actually the sales. So together, orphan drugs for orphan indication or for non-orphan indication made more than $100 billion. That's good. So the orphan drug research is also going at a rapid pace. So my talk is focused on paradigm shifts. What are paradigm shifts? Where do we need these shifts? The thing is, technology is evolving. That is the best answer I can give. A traditional drug discovery before 1960s or up to 1980s typically used only animal models and going into the clinical trials. Today we have many other technologies that we apply for drug discovery, but this is phenotypic drug discovery, which means you're looking at symptoms of a disease and see if your drug candidate actually subside those symptoms and make it as a drug. That is what simply phenotypic drug discovery means. But in those 1960 through 1980s, if you can see that any drug that went into the humans, 40% of them failed in phase one trials. They don't have the pharmacokinetics, desirable pharmacokinetics, to be effective as drugs. And the other one is efficacy. Efficacy, many of these drugs do have 30% success rate out of 100 drugs. 40% fail due to PK, and another 30% be successful for efficacy. And if you look at the animal talks, about 11 to 12% used to fail. And also there are other issues, that adverse effects in man that you didn't see in animals. And there are other reasons for commercial reasons and manufacturing problems. Also, drugs are not approved. So what happened was, in that phase, 1980s to 1990s, there was a lot of innovation that happened in PK, PD modeling. And also we have a lot of molecular biology tools, and we understand the disease process much better. So what we have is, in 1990s onwards, a targeted drug discovery. We have specific enzyme receptors or enzymes or other targets. I mean, you pick them up based on the molecular pathway of the disease, and you try to inhibit that particular enzyme with your chemical modifications and to see. And in fact, molecular modeling today applies to that area. And in fact, you do the molecular biology of the disease process. At the end, you look at the 1990s, when you come and look at the chart again. In fact, it didn't really change much. It shifted from one paradigm to another paradigm. Efficacy is really unchanged. 30% before 1980s is about 36%. I wouldn't say that's a great improvement. But the problem is with the increased pharmacokinetic in the clinic, these drugs, or 43% of them had died because unforeseen toxicities. These toxicities, they didn't see them in the animal models. And that's the most important part is the animal models really didn't translate into the toxicity. So most clinical failures happened due to these safety issues in 1990s. And PK failures have gone down to 14% only. So you can see that it's shifted from one area to another area. So the pharmacokinetic failures now shifted to toxicity failures. That's happened in targeted drug discovery approach with improved pharmacokinetic models. Then you come into the 2000s. Now we understood that the diseases are not just simple diseases. Any disease has subtypes. If you take breast cancer, there are about 20 subtypes due to genetic mutations and other things. So we are not born equal. Everybody is different. In this seminar, if you look at all the people that are attending here, if there are 100 people, I'm pretty sure about only each drug may be working only in 20% of the population, not in 80% of the population. The reason this is very important is because when you're going into the phase 3 trials, you need to stratify the population. Today, the way clinical trials are done is that all or none effect. You take a lot of people who have the disease and try the drug and see if it works or not. Miserably, they fail in the phase 3 clinical trials. But if they stratified the population in the phase 3 clinical trials, only pick the people who will be responsive through a genetic biomarker, those studies are much more successful than just taking all the 100 people in the disease population and trying to dose the drug to them. So now we learned a lot. That's called personalized medicine. Now we are also going into the precision medicine. As I already told you, the animal models are not really translating into clinical stuff. But if I go to regulatory agencies, they have recommended studies, mandated studies, I would say, that you need to do toxicology and pharmacology before you go into the clinic. This is very important. Even after clinic, there are lots of toxicology studies done, long duration toxicology studies are done. They have to be done by book, mandated by FDA. So with that, we are still having more issues because regulatory agencies are not changing their paradigm. They've been using the same paradigm they used in 1980s, whereas we in scientists and industries have moved. Moved a lot to with the new technologies and other models, trying to understand the science better. But regulatory agencies still recommend animal models, although they don't work very well. So that is another paradigm that we're going to be moving into. We are currently in the process of moving into now. So where is the problem? The molecular complexity in drug discovery is that we know that every human being may have about 20,000 genes in their cells. We have 3 trillion cells or more. I don't know. I lost the count recently that. But 20,000 genes. Do we know all the functions of all these genes? No, we don't. But interestingly, that liver doesn't express the same genes as your heart expresses or lung expresses. If they all express the same genes, they won't look like lung, liver, or heart. They look like the same. All of them look the same. So we don't know the functions for all these genes. Not all genes are expressed in all tissues. And most importantly, inter and intra-ethnic genetic polymorphism of many genes. Many genes, if you actually sequence them and you find a lot of mutations from the wild type gene, and in fact, some genes, there have been at least 100 to 200 mutations, some genes. But many other genes are also conserved with only 5 to 10 mutations. So that's another complexity. There's another thing is that we age. During age, we also have susceptibility for gene mutations due to the environmental pollution or the dietary lifestyle factors and many more issues that we have. We accumulate gene mutations. And those gene mutations are age dependent. At 30-year-old, you may not be susceptible for cancer. Maybe at 45 years old, you are, because you've accumulated new gene mutations that may actually put you into the risk factor for cancer. Take the protein, right? Proteins, there are 1 million proteins. I still don't understand the concept, because it used to be one gene, one protein hypothesis, but now we know that it's not true. So there are more proteins because of the gene splitting, and so the same sequence of some part of the gene also works with another gene at the sequence. So this is how proteins are too high in number, 1 million proteins. The other problem is that the proteins as such, from gene to protein, sometimes doesn't work that way, because there are post-translational modifications on the proteins. And many of them are also membrane bound. Membrane bound are conformationally different from purified protein. And family of proteins work together. In some genes, what this is a gene family, 3 or 4 genes with 3 or 4 proteins work together. And those are not one gene protein that you can work with. So it's not. And so the family of proteins work together. That's another complexity that you have. Epigenome, I would say it's like a space map. We don't know. We still don't understand what it is, because there are lots of transcriptomes, microRNAs, long coding, non-coding long RNAs, siRNAs. There are many other factors that we don't know how they regulate the gene function and protein function. This is still understanding in more and more details, but we are getting there slowly. And the final is metabolome. Typically, I would love to look at the genome to phenome, which is the metabolome is the phenome that you can actually look at, measure them in terms of the whether gene mutation has happened. You can look at the metabolite and see if that metabolite is there that for that particular gene or not. But again, most of the metabolites that we know today are at quantifiable level, only about 7,000. I think there are maybe many other metabolites that we cannot even see in our biological menu, because we don't have sensitivity techniques to go to zeptogram levels to see those metabolites. They are very transient, they disappear in milliseconds, and so those metabolites circulate below quantifiable levels, so we don't know how many are there. So this is the complexity that we are dealing with when we want to discover drugs. On top of it, we also have lifestyle diseases, right? And we know glucose, if glucose goes up, you get diabetes. It's a natural molecule that is very important for you to get energy. And glucose is the source of energy production, ATP production in our cells. And then, cholesterol. Cholesterol is also very important for biological functions. But increased cholesterol has been associated with heart disease. Phenylalanine is an amino acid, and you know that there's an enzyme called phenylalanine hydroxylase. If it is deficient, it causes phenylketonuria. As the phenylalanine in the body accumulates in these people with phenylalanine hydroxylase deficiency, they get serious defects. These kids who are born look very healthy, but after a year, you see their health is deteriorating, mental problems, cognitive issues, and also the physical issues as well. And the important thing is oxygen. Oxygen is not really biological component, but it is an integral part of biological system, because during ATP production in the mitochondria, oxygen is very much required. Oxygen has four electron reductions converted to water, which is a very important process to get energy in our bodies. But in sometimes in disease conditions, oxygen actually gets one electron reductions, and through which you have superoxide radicals, hydroxyl radicals, singlet oxygen, etc., which are very damaging to mitochondria as well as to the other subcellular organelles. Triglycerides. Indians are more susceptible to hypertriglyceridemia. In fact, the hypertriglycerides actually causes pancreatic disease, and this is the pre-diabetic stage. And this pancreatic disease actually fools the liver and muscle, saying that there's not enough glucose. So what happens is uh, you try to get more glucose converted to glucogen and glycogen, and there's a huge problem there. And triglycerides do cause as a pre-diabetic effect. And I've published some review articles also on these processes and how the natural poisons in the body, endogenous toxins, I call it, do contribute to the disease processes. So let's go back to drugs, right? And typically we like to give an oral drug. I mean, everybody wants an oral drug. Man, it's easy. I mean, if I have a oral drug every day, I take it. I don't forget it, and it works very well for me. People have used this like statins every day for 7 years to 10 years, or even lifelong. They've used it. That's very good. Every day you take one pill, you don't forget it. But if I have a drug, it's not oral. If it is injectable, and I have to go to the doctor. I mean, especially if you have to do intramuscular injection, you have to go to your doctor. I can't do it myself, or nobody can do it. So you have injectables also pain, and nobody wants to take a needle into their body, and that's another reason. But even oral drugs, if I have a drug that has to be taken once a month or once a week, I tend to forget it, right? Anybody will tend to forget it. So you're not complying with the drug dosing regimen. And that's the non-compliance of drug dosing. It's not nothing to do with the compliance of the your other activities, but you're not taking drug routinely as recommended by the doctor. So that's why you prefer oral drugs over the skin or other routes of administration. Now, drugs have to go into the intestine after you take a pill into the intestine. You can see that there's a gatekeepers there. They're called transporters, especially efflux transporters. And these transporters don't allow drugs to get into the body. Many drugs don't enter unless you give high dose to saturate these transporters. So when you do that and these drugs don't get in and you increase the dose to do it, but increasing the dose may have other side effects. In fact, many of the drugs also, if you have herbs, cytokines actually downregulate some of these transporters. And they also inhibit the transport. For example, there's a herbal drug that we know very well in grapefruit juice. It's not a drug, but it's a chemical that actually can inhibit these transporters, and more drug gets into the portal circulation. So what happens is with that, you have a dose dumping in the intestine, which is another problem there. So the drug, once it's absorbed in the across the intestinal wall, it goes through the portal vein into the liver, and liver is another barrier there. It's a screen. It doesn't want any foreign material to go into your body. So, for example, drug metabolism enzymes chew up the drug, and they conjugate them, they make them more water soluble, and they throw them out of the kidney. And this happens to many drugs. So we need to understand, are our drugs stable from drug metabolism processes? And that we need to look at before we go into the clinical trials. The other problems is that these liver drug metabolism enzymes can be inhibited by other drugs. They can be downregulated by the cytokines or other herbs. And they are also inducible. Some of these drug metabolism enzymes are controlled by nuclear receptors, and these nuclear receptors actually increase the activity of this is a part of detoxification process. This liver wants to get that drug out of the body, so it increases the enzymes of these drug metabolism enzymes, tries to clear them from the liver and your body. That is the process that this liver does with these enzymes. And sometimes what happens is that these drugs, especially due to drug-drug interactions, one drug inhibits the metabolism of other drug. The one drug gets high concentrations. The high concentrations go to heart, and they stop the potassium channels, which are required for the heartbeats. So they cause arrhythmias, and some of these arrhythmias are fatal. And many of the drugs that are actually marketed were also withdrawn later because of drug-drug interaction issues that were not predicted before they were marketed. So drug metabolism has several other consequences. Uh, Not only drugs, these cytochrome P450 enzymes in liver, they can metabolize environmental pollutants as well. And due to the process of drug-drug and drug-herbal interactions or drug-drug, etc., these are modulated. Drugs can also be metabolized to highly reactive free radicals and electrophilic metabolites. They're very damaging. You don't see them. If you take the blood sample and look for the metabolites, you don't find these metabolites because they're bound to the proteins and DNA, covalent binding. And that is actually can cause immunotoxicity. It's like a hapten model. A small molecule binds to a protein that actually causes immunogenic reactions. It becomes an antigen too, and they produce antibodies against that also, that protein. The other thing is the detoxification process. As I said, phase two metabolized sulfation, glucuronidation, etc. are there. These are detoxification processes, and all these processes compete with each other in the liver. This is primarily. So how do we analyze these things? Typically, if I give a drug orally, I follow the blood concentrations of the drug and see how much drug is there over the time. Like look at the time versus plasma concentration of drug curve. If it is IV injected drug, it's a straight curve, it's going down. But if you give an oral drug, because of all these barriers that I have in the intestine and the liver, the absorption is going to be much less compared to the orally injected drug. So in this case, I would say the bioavailability is only 10% of the drug. Sometimes some 10% bioavailability is okay, but ideal bioavailability is about 25 or higher. That's what we look for. The half-life also, we look for a drug. Ideal half-life is about 8 hours. If any drug has a half-life of over 20 hours, unless you're giving once a week dose of the drug, we don't want to take for a daily dosing that kind of a drug into clinic. And there's also a lot of gender and other variabilities. In this case, I'm just putting a gender variability of the clearance rates. And you can see the males versus females, there's a huge gender difference. I mean, two times the higher clearance in females compared to males, if you average that up. And so there are various factors contribute to the variability from person to person also. I'll come to that later. Genetic factors also contribute to that, the variability. The major issue that we face is the nonlinear pharmacokinetics. Nonlinear means that if a drug is not metabolized, it's not a very good transporter substrate, and I expect a linear pharmacokinetics over several doses, like 50 to 1,000 milligrams doses per kilogram dose. I usually get. So that's why we need to know if this drug is metabolized, if this drug is a transport substrate or not, before you put it into the animal trials or in the clinical trials. So if you have a nonlinear pharmacokinetics, that complicates your dosing regimen. You don't know how to dose this compound. What dose is good for one person versus another person? It's a big debate about it. We don't know many times with nonlinear pharmacokinetic drugs, we don't take them into the clinical trials. So here say that there's a linear PK, there's no brainer, we'll take it forward. But if it's a nonlinear, one is a PK dependent, what happens is metabolism saturation or something else, transporter saturations, they get to nonlinear phase after 500, after 250 milligrams per kilogram dose. These are arbitrary doses I selected here. And if you look at nonlinear PK2, in this kind of cases, sometimes the drug itself inhibits its own metabolism, or it induces its own metabolism. It can happen both ways. And if it does, the pharmacokinetics change for the same drug up on multiple doses. And these are some of the complicating factors when we go into the clinic. So we need to understand these processes before we go into the clinic. The cytochrome P450s, it's been done. I think it was discovered in early 1950s, this enzyme. And there are a variety of these enzymes. Family 1, 2, and 3 are the major ones that metabolize drugs. But if you look at the species differences, mouse, rat, dog, monkey, and human have different uh, enzymes. If you look at the 2C enzyme, there are about a dozen 2C form isoforms in mouse. Compared to humans, there is only 3 isoforms, and even in rat. So when I'm taking rat as a model, if the drug metabolism is happening by 2C in rat, I don't know how that I have to relate it to the human situation. And these are the problems when you use the animal models, the mouse, rat, dog, because of the species differences in these enzymes, which catalyze different substrates and also have different catalytic properties that complicates it. In any case, in humans, we know that CYP3A4 is the major drug metabolizing enzyme. If you take all the marketed drugs to date on the market, now 50% of those drugs are metabolized by CYP3A4 and followed by CYP2D6 in humans. It's about 30% of the drugs are metabolized. CYP1 and CYP2 are more important in environmental pollutants and pesticides, etc. But those enzymes don't contribute much to the drug metabolism. You can say caffeine, maybe 1A2 is also metabolized by, sorry, caffeine is also metabolized by 1A2, 1A1, etc. And if I have very high levels of 1A2 enzyme, and if I drink coffee, I don't have, I mean, I can sleep right away because the caffeine is metabolized and excreted out, and it doesn't have any effect on my sleep schedule. Uh, if you look at the variability in a liver, if you look at the CYP1A2 enzyme versus the 3A4 and 2D6, the variability in the liver, if you take 100 human livers, right? The variability is about 60-fold for 3A4. Okay. 2D6 is only 2.5-fold difference. But if you look at the variability in terms of the catalytic activities of these enzymes, 2D6 has 1,000-fold difference in the catalytic activity in the individual humans. I mean, if you take 100 individuals, you can see a few people, 10 people or maybe 20 people may have 1,000-fold higher catalytic activities compared to other people who have normal activity levels. But 3A4, in spite of having 60-fold variability in terms of the total content out of the in the liver, its variation in the catalytic properties is only 90-fold. So these are very important in terms of the selecting a dose for a human to human person to person as well. I already said transporters are also very important, and transporters have a lot of endogenous substrates as well. I'm not going to get into the endogenous substrates, but if you look at the intestinal walls, there are efflux transporters on that side of the gut lumen, and also on the blood side. There are transporters also efflux into the blood side. So there are quite a few transporters out there that we need to deal with to understand, are these transporters the substrates for the drug I want to take into the clinical trials. Then you can also see the liver hepatocyte. If you look at the hepatocyte on the canonical side, you can see that there's several drug transporters, and also on the blood compartment side, there's others. So from the blood, it's getting uptake transporters, and it has to be effluxed into the bile through these transporters on the biliary side, canonical side. And so you have also the brain. Brain is a big barrier. Many drugs don't go into the brain. And interestingly, some drugs actually penetrate very well into the brain, although they work very well for your disease, but they cause toxicities in the brain. So we also make sure that if it is a drug that we want for uh, uh, heart disease or something, we don't want that to go into the brain and damage the brain cells. So we also look for negative effects in such cases. But if we want a drug that we're looking for glioblastoma, brain cancers, or other brain functions, the neurotransmitter functions, etc. We want that drug to go to the brain better than the in the blood circulation. So it has to penetrate very well. So these transporters play very important role in brain also. That we look at these, any of these transporters have any problem as a barrier to get the drugs into the brain. I told you that there are intraethnic and interethnic variations in CYP26 gene polymorphism. This is one well studies cytochrome P52 in humans. Uh, there are so many mutations. I think there are, I lost count now, maybe 150 mutations. But the STAR4 mutations are the most important one. This is about 10% of the Caucasian population are mutated with, and so the enzyme is not present. You can see that uh, it changes the drug metabolism in 10% of the people populations. The same STAR4 gene in Asians is about 20% variable. And so 20% of the Asians, especially Chinese, Orientals, are there actually 25% deficient. So if you're looking for a drug dose with the CYP26, you need to do an ethnic population dosing curves, because Chinese don't accept the same dose as the Caucasians. Even in Caucasian, 10% don't accept the same dose. So African Americans have a different mutation called STAR17. And that actually is hypermetabolic, and many Africans actually don't get activity with the same drug that works very well in the Caucasian populations. So the ethnic diversities have to be considered when you're doing, especially with CYP26. You need to make sure that this drug works across all the ethnic groups. Okay. So as I said, there are also induction aspects of it. If a drug is given, it goes into the liver. There's a pregnant X receptor. It's a nuclear receptor, and it hybridizes with retinoid X receptor, and that goes and binds to the uh, pregnant receptor element on the gene, and that activates it. And you have, especially with this PXR, you have multitude of transporters, phase 2 enzymes and cytochrome P450s elevated their concentration. This process is for detoxification. So the more enzyme is there, the drug will be removed from the body very fast, because the body is considering that is a foreign material, not something useful for the for our body. So problem with this kind of mechanisms is that drug that doesn't work in many people. So in fact, I say many drugs actually work only 20% of the populations. They don't work 100% of the populations. So you need to do population stratifications with these models. The other issue with cytochrome P450s is the inflammation is a major detriment. Healthy people have a normal immunology, immune cytokines, etc. But people who have diseases, they always have pro-inflammatory cytokines. These pro-inflammatory cytokines actually inhibit or downregulate, not inhibit, they actually downregulate the expression of cytochrome P50s. So if I do my clinical trial with a healthy volunteer, and if I look at the pharmacokinetics of the drug in a healthy volunteer versus the pharmacokinetics in a diseased patient, is going to be totally different. So I have to come back to the square one. So in fact, when we go to the phase 1 clinical trials, according to the FDA, we must do with healthy volunteers. We don't want to put any drug into the diseased patients unless it's an oncology drug. Typically, oncology drugs have some waivers that we could do it, but cytochrome P50 enzymes are downregulated, many of them, in so they're not expressed very well in diseased populations. And so that has to be considered when you're moving from healthy patient to the healthy subject to the, is not a patient, I'm sorry, it's a typo, healthy subject to the patient, okay? Uh, so what happens is you have to compare the drug metabolism in healthy versus the unhealthy. Also, that many drugs were marketed and they didn't have any problems in the clinical trials. They didn't have any problems in the animal trials. They were, for example, troglodyzone. My friend Dr. Uday Saxena, my collaborator at Rage in Biosciences, extensively worked when he was at Park Davis on this molecule. The interesting thing is this troglodyzone doesn't have any effects in the clinical trials, hepatotoxic effects. The reason for that is because the troglodyzone dependent deaths occur in 1 in 5,000 people. 1 in 5,000 people. Clinical trials are not conducted not more than 5,000 patients. So how do you pick up these idiosyncratic effects in 1 in 5,000 people effect? But if you let 1 in 5,000 people, and if million or 10 million people are taking the same drug, you see a lot of deaths. That's what happened to troglodyzone. After it is marketed, they saw probably 5,000, 6,000 deaths in 1 year. I don't remember the numbers, but the Park Davis was asked to pull it out of the market by FDA. It was making $2 billion a year by then. You imagine you spend $2.5 billion to get a drug into the market, and then it was making $2 billion, but you have to pull it out of the market because of the serious side effect. And now the patients' families who have died are actually suing the Park Davis or any other company who has this problem, and you have to pay $5 billion in lawsuits. So it is really risky business. I mean, I don't want to go market a drug that will kill people at the end, but which I could not prove it in clinical trials or in animal trials. These are some of the major issues that when the drug development actually has major risk factors, it's not easy to do it unless you understand. But every drug is different, unfortunately. So you have a number of drugs like this, hepatotoxicity, were withdrawn over the years. To date, I think out of many drugs, 450 plus drugs have been withdrawn from the market that with unforeseen adverse events or toxicities. In fact, there are some drugs that are now being considered for withdrawal. I'm not going to discuss, but these are also causing hepatotoxicity in the market now. In fact, another problem is the cardiac toxicities. Heart is the most sensitive organ. As I said, drug-drug interactions is one of the serious considerations to be given. Until 1990s, nobody realized it. Since mid 1990s, about half a dozen drugs showed problems in human populations. Many patients are dying suddenly, and that was related to the drug-drug interactions. Anybody over 50, 55 is probably taking 6 to 7 drugs or higher because they have series of disease problems, not just one disease. They have They just don't have pharmacovigilance to understand what are the interactions with these alternative medicines with the drugs that we are taking also. Nobody knows about it in India, that alternative medicines can actually interact with the drugs and can kill patients. There's some of these drugs, or most of them are antibiotics, are listed here. And many, if Posicor is one of the drug that was marketed by Roche in 1996. Within 1 year, about 3,000 people died with Posicor after it's marketed. Then it was realized that 16 other marketed drugs interact with Posicor in drug-drug interactions. And there's a serious fatal cardiac interactions. Your heart suddenly stops. This has nothing to do with cholesterol. This has nothing to do with your other heart diseases. This is basically what it is doing is stops your heartbeat and stop your heart. And many times people die, and I do wonder that they're taken to the crematorium, and I wonder, did this patient die of some drug-drug interactions or drug-interval interactions? Unless we do pharmacovigilance, we'll never. So I would say that people, families, whoever their patients died, who are taking many drugs or many herbal medications, they should have a post-mortem to see if their deaths are related to the cardiac toxicities with drug interactions. The other complexities is in the disease mutation, gene mutations in the disease families. We know about breast cancer, right? Breast cancer, inheritable gene mutations, there are about that are listed, quite a few of them. And there are about 5 to 10% of the population breast cancers have these gene mutations. And unless you identify these gene mutations, you don't know which drug to give. So there are some drugs have already been developed for some of these mutations. So this calls for you need to know what gene mutation this breast cancer patient has, so that you can appropriately treat him. Unless you do that, you cannot not just give any drug to this patient and expect him to survive for a long time. And also, researchers identified about 110 genes associated with breast cancer. 110 genes just identified, but mutations could be as many as 400 mutations or 500 mutations. In fact, 33 breast cancer risk loci have been identified recently in 2018. So this is getting more complicated. It's not a homogeneous disease, it's a more heterogeneous disease. Breast cancer is not a homogeneous disease. And you also have environmental risk factors, tobacco smoke, dietary, excessive alcohol consumption. Interestingly, that many of these things, that especially some of the pesticides and dietary estrogens, these are hormonal imbalances which cause especially women, because the hormones are very important for the women. And if you have these hormonal imbalances, they cause breast cancers because they increase the inflammation and other things in the breast cancer. So it's a multifactorial, many risk factors are involved, it's a heterogeneous disease. Same thing with autism. A lot of people think autism is one disease. It's not. Researchers have identified 102 genes linked to autism, and that actually has more than 800 gene mutations identified. Okay, now I have to identify 800 gene mutations. Diagnosis is going to get very complicated. You have to develop the tools for all these 800 gene mutations before you discover a drug for this autism treatment, right? It affects in 1 in 59 children. So we don't have really diagnostic methods for autism, really to treat these autism kids. So what we are doing is with all this complexity of the molecular pathways in humans, and every human is different, we are changing into a paradigm shift into personalized and precision medicine. Every human in the future will have his own gene chip that can be scanned, and based on the gene script, and also protein script that will be also available maybe in a decade from now. Using that, you actually give what drug to give to the patient, what dose to give to the patient. This is the futuristic, maybe 10 years from now. I hope so. I mean, that we would have it. But what are the other problems? As I said, we don't have animal models, right, to really understand. So one thing is you need to develop molecular diagnostics tools. This can be done with whole genome sequencing, both including the exome sequencing also. This is out of the mutations. I mean, between the genes there are other sequences that are promoter regions that you also need to sequence them, because gene mutations alone doesn't tell you that there's a problem. Sometimes the promoter regions are also mutated, or regulatory regions you call them. Non-animal human 3D organs. That, in fact, Reagent Bioscience is really trying to get this technology up and running, because our future goal is to use different organs and combine them together. It's like a human, mini human on a microchip. to evaluate the drug screening process or drug development process before you go into the clinical. And the other thing is you also need to know how the drug actually travels from one organ to the another organ, which organs it accumulates. So you need a model because you cannot directly go into the humans and do all these things. It's very expensive. First of all, you don't know, you cannot take out all these tissues. In animals, you can take out tissues and measure the drug concentrations, but in humans, you cannot do it. Yeah, you can take blood and do it, but if the drug is actually accumulating in thyroid, you cannot take thyroid and look at the tissue concentrations. So for that reason, you need an in vitro human model that can be applied like an animal model to understand the human uh, based diseases, how they do it, and stratifying the clinical trials with molecular markers for a successful drug discovery. This is already started, but is not still into the world practice in the clinic. Very rare cases, some people, some diseases they're doing it, but very rare it's happening. Now you need to understand which patient has which mutation. So you take the blood sample and screen their genes. You can do that with some genes. Hello, hello, hello.
Yes, Uncle, we can hear you.
My screen gone.
But we can see your screen, sir.
Okay. Sorry. Yeah, got it.
Yeah.
Then the other thing is that because we have all these issues, I mean, taking a drug into for 15-year clinical trials and path to get marketed is a huge process. I mean, many of us live maybe 60 years. I mean, you will start working at 30 years in a pharmaceutical companies. In 30 years, we may be able to discover 2 or 3 drugs maximum. That's it, the strength. But if you're working in a team environment in big pharma companies, you may be able to discover more drugs. But individually, we can't do it. The other thing is the drug repurposing. There are already 8,000 FDA drugs are out there in the market. What we are looking now that are these drugs can work for other diseases, not just for one indication. And this is where I think you can condense the timelines. Maybe you can, with the drug repurposing, even there are lots of challenges, but you can condense the overall drug discovery and development process to. There's no real drug discovery there. It's a more drug development process for the clinical side. You do have very limited drug discovery process there. And you see if you can invent this different therapies for the drugs are already marketed. There are already a few drugs out there in the market. You guys are knowing with the COVID in the last 2 years, people are using hydroxychloroquine, ivermectin, many other remdesivir. Remdesivir is actually approved for Ebola therapy, but now they consider that it could be used for. So there are lots of other drugs, ticlozimab, which is uh, used for normally for I think arthritis or some other diseases. All these are drugs are repurposed, but the problem is none of them actually made an impact because hydroxychloroquine has a lot of drug-drug interactions, and ivermectin has limited uh, efficacy profile. It doesn't really work in everybody, but if you give oral tablet, how are we sure that actually COVID is a respiratory disease? Oral ivermectin, how do I know that it actually goes into the lungs and inhibits the coronavirus? So these are practical problems people don't think through. They just use it and then they realize that the data is not convincing that it really works for. And FDA and other regulatory agencies and come say that don't start giving these hydroxychloroquine and ivermectin. There are other issues. We don't want that. And these are the issues that drug repurposing challenges are. That so we need to understand how it distributes in the humans, what kind of a dosing therapy. If the ivermectin and hydroxychloroquine are given by nasal spray, spray therapy, nasal mist therapy, or something, maybe they would have worked better. But so far, if you want to do those therapies, then you need to do the clinical trials again, because when you change the route of the administration, you need to do separate clinical trials for that particular route of administration. Now, with all this paradigm shift that is ongoing right now, what do we face? What kind of roadblocks we face? For example, regulatory is too slow to catch up with scientific advances. And this has been age-old problem. We've seen from 1980s. They still recommend the same animal models which don't work. So we don't have to choice. We don't have any choice. We have to do those animal trials just to make FDA happy, or European what is that, agencies happy, and these regulatory agencies are not catching up with the new tools that are happening. But I think now they are realizing it. They are ready to use these human alternative models and mandate them in the near future, maybe 2025 or further. Lack of research funding sources, especially in India. I mean, venture capitals and angel investors are looking for a quick exit. So if they say, I'll give you $1 million today, can you give me $2 million tomorrow? This is research, drug discovery research. It's not something I'm selling toys on the street, right? So these are risk associated. So we do need funding. Uh, that risk venture capitals and investors should take to improve the research funding in India to be able to do. For example, in the US, if anybody has a novel idea that they can try to cure some particular type of cancer, and many people do get $50 million venture funding, even though they don't have any proof of concept, they don't have any revenues in their pocket, right? So the other problem is that every day I face general public who read Google junk, and they come and question my scientific integrity, my scientific knowledge, and say, "Hey, you guys know anything because you guys can't discover drugs." I think we should move out of this kind of mindsets. People are getting 5 years, 7 years, they're spending degrees and working in premium institutes in the world and doing this research. There are lots of problems in discovering drugs. So we need to, the public also has to be supportive of this research, not ridiculing this research. Lack of collaboration between academic researchers and biotech pharma industry. This is very good. If you go to the Boston area, MITs, Harvards, and even Princeton, and everybody there interact with small biotech or even big pharma industries there. And in fact, lots of academic research, the actual drug discovery happens in academia, not in. We take that target, whatever they discover, and we validate it and see if it can be a target for some treating some cancers, etc. So we industry evaluate it and do all the clinical trials that are required by the FDA and others. So that requires a lot of money, and we raise the funding for it and try to get this drug into the market. But academia is very important and crucial in partnering with industry to do this drug discovery research. And I mean, I'm telling you guys who are all their academic participants in this, think about it when you go into tomorrow into a company or something that you want to do. Promote academic and industry collaborations so that we can do cutting edge research in India. So I said that what is this human organ in a chip? This is not ready yet. I mean, basically, it'll take a while for you to build this model. What we can do is we can measure all parameters, even drug concentrations and histopathology in the tissues. If we have a model like this, it's more ambitious too, and there are lots of microfluidic chips. In fact, Dr. Saxena in our group is actually building 3D bioprinting tools to do the organ-organ interactions. But these are not just done in one day. You need a lot of funding. I mean, maybe minimum 20 crores to set up these multi-organ human chip. And we want to combine that physiologically based pharmacokinetic modeling so we can predict the human uh, pharmacokinetics, human disease effectiveness, efficacy, everything using this kind of model. But this is where the future lies in if we still don't want to play with these animals and want to get into the uh, and now, of course, artificial intelligence is also making a lot of. If we plug in the known parameters of the human physiology and use this artificial intelligence and to predict the human efficacy and toxicity parameters, that would be a very good boon for us. And in fact, maybe 90% drug failures, we could come down to maybe 50 to 40% drug failure. That is a doubling the efficacy and making more drugs into the market. Instead of 40 more drugs into the market or 50 drugs into the market, you may go up to 100 drugs into the market every year. And this is what we need in the future. So with that, I'll stop it. And in fact, my favorite quote from the Steve Jobs. We have to be creative. We can't just do routine work and expect results out of it. You keep at it. You know what? Knowledge, we all have knowledge. You know, these pieces of knowledge that we have, they're everywhere. But we don't know what that knowledge means, how to make it productive in terms of the creativity and come up with a drug molecule. Yeah, many of us also have experience. We can connect these dots in in a different fashion, but we don't see anything out there. There's no relation there. But Steve Jobs says there's a creativity part can identify these dots to a cat. You see the cat in there, if you're a creative person, then you're a genius. And I think that's what we need to be doing, not just hard work and not just knowledge. We also plug in the creativity to be more successful. And I mean, I know these guys have applied those tools for software industry. We could apply those tools into our drug discovery research as well. Thank you.
Thank you, sir. That was a very insightful talk. I think the information and the insights that you gave today was it showed us a very interesting side of the drug discovery process that we hadn't seen before. So, following that, we have a few questions that the attendees of today's talk would like to ask you. Can I go
Yeah,
ahead with that?
yeah, sure.
Okay. So, the first question we received was, when does a drug follow linear pharmacokinetics? Is there a specific um, factor to it?
Sorry, I didn't get your question. Can you say it again? Sorry, you were breaking
Yes,
up
sir.
in.
Sorry. So the first question we received was, when does a drug follow linear pharmacokinetics? Is there a specific factor to it?
Yeah, as I said, metabolism, drug transport, these are the critical factors that alter even protein binding. I mean, many drugs are transported by the proteins. They may not follow the linear kinetics. So, and in fact, there is a saturation pressure. If these drugs are not get saturated, they don't get metabolized anymore. They don't require the transport processes. They don't require protein binding processes to be transported. Then sure, the linear constant. But the problem is when you go to that linear concentration, you're at very high doses
Correct.
that can be problematic. They can cause serious side effects. So you need to be able to drugs to come into a linear pharmacokinetics where the drug is effective for the disease with minimum manageable side effects. That's the reason we actually screen for drug metabolism, drug transport early on before we go into the clinic and choose the drugs that don't have these liabilities.
Okay. That's useful, sir. Thank you. The second question was, what is pharmacokinetics based on? Are there any phases for drug addiction in our body?
Uh, Repeat it again, sorry.
Yeah. What is pharmacokinetics based on? I think he meant the principle. And are there any phases for drug addiction in our body?
It depends on the type of the drug. For
Okay.
example, in cancer pain, typically the drugs of choice are opioids,
Okay.
oxycodone, hydrocodone, codeine, etc. The drugs that are given, morphine for the cancer patients because they numb your nerves and you get pain relief. But the problem is some population of these patients do get addicted. With the addiction, what happens? The problem is they'll have withdrawal symptoms. What you do is after a while, hey, this patient is fine. I don't need to give this drug anymore because you may get addicted. But you withdraw the drug, and he will ask you for the drug every day. I want the drug. I want the drug. He's addicted to it. There is some precedence that addiction is also genetic. For example, there are reports that in oxycodone, 20% of the cancer populations get addicted, and there is apparently the gene mu receptor, which oxycodone binds to, as 20% of the people have one particular mutation. Those are the ones that get addicted to. But apart from that, there need more studies to tell that. And so the addictive drugs are different category. They're regulated drugs. They're not they have to be prescription drugs, although they are generic drugs. So also the anesthetics, some of the anesthetics are also addictive, and depends on these type of drugs that you have to be careful for addictive liabilities in treatment.
Okay, thank you, sir. I think that answers the question. The next question is when is it known which human cytochrome P45 enzymes are involved in metabolism of a compound?
So what we do is we don't go into the humans. We take the human livers. Today it's still not reality in India yet, but in the U.S., if anybody dies on the road by an accident, I have my driver's license in my pocket. I can write on that in the back of the side that my organs to be donated. Then they use that and they actually take the organs of the dead person and they actually distribute to the research organizations like us. And we actually take those livers and we prepare the subcellular fractions from those livers, especially the endoplasmic reticulum, which contains the drug metabolism enzymes. And we actually store them in -80 freezers and use them. And also, the other technique is cryopreserved human hepatocytes. You can store these cryopreserved human hepatocytes and use those instead of using subcellular organelles. You could even use these cryopreserved hepatocytes. Those are the tools that we use to identify which P50 enzymes, which phase 2 enzymes, or other enzymes metabolize these drugs, and would they have any liabilities for drug-drug interactions, or would they undergo inductive drug metabolism disposition, or these drugs inhibit those enzymes and cause drug-drug interactions. So we do that extensive research before we go into the clinic to understand about these drug properties, whether they have serious liabilities or not.
Okay. Okay, thank you, sir. The
But in
next
India,
question.
what they do is they import those tissues, for example, Syngene or Dr. Reddy's and many other of these research companies in India. They import them because we don't have a source of getting these human livers in India.
So I think that's something that we have to work towards
Yeah, right,
to
exactly. Yeah.
improve our research. Correct, sir. The next question is what are the advances in understanding the effects of diseases on pharmacokinetics, particularly changes in the metabolizing enzymes and transporters with gene disease progression? Should I repeat it, sir?
It's an evolving field. It's an evolving field. We know a lot about drug metabolism in the last 30 years. We characterized all the P450s, the subtypes and everything. But transporters are still, only a few transporters are characterized. We are still characterizing many other transporters. So we still don't know enough of transporters. So because these drug metabolism, enzymes and transporters work together. So we need to understand them better together. And that research is going on still. We're waiting for more data for it.
Okay, sir. Okay.
Yeah.
There's one more question from Uma Purnasamy. Her question is for you to provide an understanding to psychiatric disorders that are caused by gene change in neurons due to the chronic exposure to addictive drugs.
It's not just exposure, chronic exposure to the addictive drugs. There are many other factors are involved. Some people don't get addicted at all.
Okay.
Only some people get addicted. So you need to identify those populations who may have addictive properties. But at the same time, the way we do it is that, if, in fact, I worked with a drug called buprenorphine. It's also an opioid type of drug, where we've actually, instead of giving an oral drug, we've started giving a skin patch.
Okay.
Skin patch delivers this drug at a sustained concentrations that don't spill into the brain. So you're actually keeping it at a level in the blood to work for your pain, in a level that it actually that also works in central nervous system. Enough goes into it to work as a pain reliever, but it doesn't go to the level of addictive uh, concentrations. So this is so you can deliver by other methods, by you giving skin patches and other methods to ensure that you're not overdosing the patient with oral dose.
Okay. So I
Okay.
think research to strike the right balance
Yeah.
is what's required. I think that's all for today
Yeah.
in terms of questions, sir. Thank you. Shresth will be taking it on from here.
Yeah. Hello.
All right. Yeah, sir. Now that the session is concluded, I would like to thank our speaker, Dr. Vangala, and the moderators, Manthan and Prithi, for such an amazing event. We would also like to thank such a lovely audience for making this event successful. The talk has been very intuitive and has given us insight into the field of drug discovery. I would also like to take this opportunity to request all the attendees to follow Research Society MIT on Instagram and LinkedIn to keep yourself updated with the events which we'll be organizing in the future. The link for the same has been put up on the chat. Thank you so much and have a good night. Thank you so much, sir. I think
Thank you
we
very
can leave.
much, Manthan,
Thank you.
and forget your name. Sorry,
Shrestha. Shrestha.
Siri. Okay. Really pleasure to be here, an honor, and thank you everybody. Okay, if anybody wants, provide my email address, and if they want to contact
All
me
right.
directly.
Sure, sir.
Sure.
Okay.
Sure.
Thank
Thank
you.
you so much. Thank you, sir.